Hello everyone. In today's class, we are going to learn about the morphology and laboratory diagnosis of influenza virus. This is an important topic for BSc MLT microbiology, especially from an examination point of view. First, we will understand what the influenza virus looks like, and then we will learn how it is diagnosed in the laboratory. Let us start with the basic morphology. Influenza virus belongs to the family Orthomyxoviridae. It is an enveloped RNA virus. Its nucleocapsid has a helical symmetry, and the virus is pleomorphic, which means it can occur in different shapes and forms. So, for your examination, remember 4 important words: enveloped, RNA, helical, and pleomorphic. Now let's understand the structure of the influenza virus. The virus has an outer lipid envelope. Inside the envelope is the viral nucleocapsid, containing the RNA genome. On the surface of the envelope, there are important glycoprotein spikes. 2 of the most important are HA and NA. HA stands for hemagglutinin. Hemagglutinin helps the virus attach to receptors on the host cell, so you can remember HA for attachment. NA stands for neuraminidase. Neuraminidase helps in the release of newly formed virus particles from infected cells. For the exam, remember this simple association: HA, attachment; NA, release. Memory trick: HA helps attach; NA, new viruses are released. Now, let's move to the most important part for an MLT student: laboratory diagnosis. We will learn this in 2 steps. First, the specimen, and second, the laboratory methods. For laboratory diagnosis, respiratory specimens are collected. Important specimens include nasopharyngeal swabs and throat swabs. Other appropriate respiratory specimens may also be used depending on the clinical situation. Remember this for your exam: influenza, respiratory specimen. There are several methods used for laboratory diagnosis. The first and very important method is RT-PCR and other molecular tests. These detect viral genetic material. The second is antigen detection, which detects viral proteins. The third is virus isolation, performed using specialized laboratory systems. And finally, serological tests may be useful in selected situations, especially when we are looking for an immune response. Let's understand RT-PCR in very simple words. The respiratory specimen may contain influenza virus. Because influenza contains RNA, molecular testing can detect its genetic material. RT-PCR helps detect and amplify the viral RNA, allowing us to identify influenza virus. There are several methods used for laboratory diagnosis. The first and very important method is RT-PCR and other molecular tests. Now, how should you write this answer in the RGUHS examination? Start with the family and basic characteristics. Then, describe the morphology and mention the important surface glycoproteins, HA and NA. After that, write the specimens and laboratory diagnostic methods. And whenever the question asks about morphology, a simple labeled diagram of the virus can make your answer much clearer. Now, let us understand the general characteristics and morphology of influenza virus. This is an important topic for BSc MLT microbiology, and the diagram is very useful for writing a short essay or long answer question. General characteristics. First, let us look at the general characteristics. Influenza virus belongs to the family Orthomyxoviridae. It is an RNA virus and is an enveloped virus. Influenza viruses are generally spherical or pleomorphic in appearance. Their size is approximately 80 to 120 nanometers. Remember, the unit here is nanometers or nm, not millimeters. Exam emphasis. So for your examination, remember, orthomyxoviridae, RNA, enveloped, about 80 to 120 nanometers. Morphology. Now let's look at the structure of the influenza virus. As the diagram appears, point to each structure. 1. Envelope. The outermost covering is the lipid envelope. This envelope is derived from the host cell membrane when newly formed virus particles are released from the infected cell. 2. Surface glycoprotein spikes. Present on the envelope are important surface glycoprotein spikes. The two major glycoproteins are HA and NA. Nucleocapsid and RNA. Inside the envelope is the nucleocapsid, which contains the viral RNA genome. The nucleocapsid has a helical symmetry. The viral genome consists of segmented RNA. So, when you draw the influenza virus in the examination, make sure you clearly show the envelope, HA and NA spikes, nucleocapsid and RNA genome. Influenza morphology, RNA, helical nucleocapsid, HA plus NA spikes. Let's look at how the influenza virus changes and spreads, starting with antigenic variation, antigenic drift. First, we have antigenic drift. This involves minor mutations in the virus. It causes gradual changes over time, and it is typically responsible for seasonal epidemics. Antigenic shift. Next, we have antigenic shift. This involves major reassortment between different viral strains. It results in a sudden, dramatic change, and it can lead to worldwide pandemics. Now, transmission and pathogenesis. How does influenza spread? Primarily through airborne droplets when an infected person coughs or sneezes, and via direct contact. Pathogenesis. Finally, let's trace the pathogenesis of the virus step by step. Step 1. Inhalation of the virus into the respiratory tract. Step 2. Attachment to the respiratory epithelium using the HA, hemagglutinin spike. Step 3. Viral replication inside the host cells. Step 4. Resulting cell damage. Step 5. Spread of the infection down to the lower respiratory tract. Continuing our discussion on influenza, let's now examine the potential complications of the infection, how it is diagnosed in the laboratory, and the methods for prophylaxis or prevention. First, the complications. These can be divided into two categories. Respiratory complications. The most common include primary viral pneumonia or secondary bacterial pneumonia, as well as otitis media, ear infection, and croup. Extrapulmonary complications. These are less common but more severe, affecting other parts of the body. They include myositis and myocarditis, inflammation of muscle and heart, encephalopathy, brain dysfunction, the rare but serious Ray's syndrome, and Guillain-Barré syndrome. Next, let's look at laboratory diagnosis. How do we confirm an influenza infection? Specimens. We collect nasopharyngeal swabs, throat swabs, or nasal washes. Detection methods. There are several ways to test these samples. Rapid antigen detection, a test that gives results in about 15 minutes. RT-PCR, a highly sensitive and specific molecular test, often considered the gold standard. Viral culture, cultivating the virus in a lab. While it can take 3 to 10 days, it is definitive. Serology, using blood tests to look for a 4-fold rise in antibody titer, which allows for a retrospective diagnosis. Finally, how can we prevent influenza? This is prophylaxis, which involves two main strategies. Vaccination. This is the primary method. We have the inactivated vaccine, which is administered annually in either trivalent or quadrivalent form. We also have the live attenuated vaccine, such as FluMist, which is given as a nasal spray. Chemoprophylaxis. This involves using antiviral medications, such as oseltamivir. This is typically used for high-risk individuals or for post-exposure prevention to stop the spread of an outbreak. This is all about influenza viruses, specially prepared notes for quick revision for your microbiology exams. For more such short notes and question paper solving series, subscribe LabTech, and our next series is about 5 marks questions from previous year question papers, 2024, RS4.